Arabidopsis thaliana, commonly known as the thailcress or simply Arabidopsis, is a small, rapidly flowering weed that is commonly used in genetic studies. The flowering time of Arabidopsis tends to vary. My goal was to determine whether inherited genetics or external stimuli had a greater influence on the plant's flowering time. According to the 1997 study Analysis of Flowering Time in Ecotypes of Arabidopsis thaliana, flowering in Arabidopsis is suppressed by a flowering locus, which can be overcome either by the genes of the plant or by external stimuli. This poses the question, are external stimuli stronger than inherited genetics in controlling the flowering time? A 2014 study conducted by H. Morikawa and M. Takahashi discusses a number of external stimuli that are known to trigger flowering in Arabidopsis. These include vernalization, sucrose, gibberellic acid, light exposure, and nitrogen dioxide. In my study, I chose to focus specifically on the effects of nitrogen dioxide on flowering time. For my experiment, I planted 90 Arabidopsis seeds in a controlled environment. The seeds were watered daily and kept under a grow light. This was my first generation. I tracked how many days each plant took to flower to categorize each as an early flowering or late flowering genotype. I cross-pollinated the early flowering plants to each other and the late flowering plants to each other. The first generation of Arabidopsis then produced seed pods, as seen on the left. I collected the seeds to begin a second generation. This consisted of 4 groups: early flowering seeds treated with nitrogen dioxide, a control group of early flowering, late flowering seeds with the same fertilizer, and a control group of late flowering seeds. In the first generation, the early flowering Arabidopsis had an average flowering time of 9.8 days and the late flowering averaged 20.3 days. Unfortunately, my second generation did not have time to flower. 3 days after seeding the second generation, I had 1 small sprout from the early flowering and 1 from the late flowering seeds, both from the nitrogen dioxide fertilized groups. In my second generation, the only germination I had came from the groups treated with nitrogen fertilizer. The control group had no germination. However, no conclusions can be drawn from the results of this experiment because of the lack of successful germination and flowering in the second generation. I can neither accept nor reject my hypothesis. To get conclusive results, I would need to repeat this experiment with an increased sample size, so there would be a larger seed sample for the second generation. Ideally, this experiment would be continued through 3 or even 4 generations, so I would need the time to continue this experiment for several more weeks.